Welcome, everybody. Hello again. Thank you for joining us. Thank you for being here for at this 68th session of the International IVF Initiative. My name is Mina Alikani, and it's my pleasure to be co-moderating this session with Dr. Mina Popovic, another Mina, and they are now calling us officially Mina Squared. I encourage you all to ask questions during this session using the Q&A section of the conference service and not the chat function, please. And of course, for videos of previous sessions, please go to ivfmeeting.com and don't forget to leave a review as well. Let me tell you a little bit about my co-moderator, Mina. Dr. Mina Popovic received her PhD from Ghent University in Belgium, investigating the impact of chromosomal instability on early human development. She's currently a researcher at Clínica Eugene in Barcelona, involved in projects relating to PGT, in vitro models of human peri-implantation development, and reproductive aging. She is a deputy of the HRS Special Interest Group on Stem Cells. And of course, her full bio is available on the i3 website if you would like to hear or read more. Mina, would you like to give an introduction to the session also?
Sure. Thank you very much, Mina. It is truly an honor to be chairing this session with you today. And my co-host is the much accomplished Dr. Mina Alikani. She is certified as a high complexity clinical laboratory director and clinical consultant. She is an editor of RBM Online and an editorial board member of Fertility and Sterility. She serves on the executive board of Alpha Scientists in Reproductive Medicine and the scientific advisory board of Tomorrow Life Sciences. Mina lives in New York and since 2019 has held the position of Senior Director of Laboratories of Northwell Health Fertility, where she runs a state-of-the-art facility that she helped design and equip. And she is now keenly providing mentorship to the next generation of ART laboratory professionals. And Mina is focused on embracing new and bold technologies, no truer than this topic today. So the topic today of synthetic embryology is truly an exciting one. This newly emerging field really seems to be taking center stage lately. So up until now, our knowledge of early human development has really relied on observing and manipulating human embryos directly. But this has in fact also been the limiting factor for really uncovering some of the more intricate processes that surround embryogenesis. So comprehensive functional and molecular studies have really been limited by the relatively short time frame that we have available for analysis, and also the inaccessibility, of course, of human embryos for research, as well as the ethical restrictions surrounding their use. So with synthetic embryology, we seem to be really rethinking the way in which we study early human development in vitro. So synthetic embryology capitalizes on this capacity of pluripotent stem cells to self-organize into structures resembling embryos. So using this approach, several groups have been able to model certain aspects of early post-implantation development, and in a recent breakthrough, even generate structures that resemble human blastocysts, which they've termed blastoids. So this new approach may really provide us with new and important ways for interrogating developmental pathways. Of course, the utility of these models will ultimately depend on how well they recapitulate the complexities of human embryo development in vivo. But nevertheless, I think with this potential, we are undoubtedly facing thrilling times ahead in human embryo research. So with this in mind, I'm certainly looking forward to this session. And now I'll pass on to my co-chair, who will introduce our first speaker.
Thank you very much, Mina. It is my great pleasure to introduce Dr. Berna Sozen. She is an assistant professor at the Department of Genetics, Yale School of Medicine, and also affiliated with the Yale Stem Cell Center. Dr. Sozen trained as a reproductive biology at Akdeniz University in Turkey, and then as a stem cell biologist at the University of Cambridge, UK. She did her postdoctoral research in Cambridge, UK, and California Institute of Technology, during which she developed an in vitro system to model mammalian early embryogenesis using various types of stem cells. Her group is now studying how embryo-environment interactions direct developmental patterning in human embryogenesis, with a long-term aim to understand the developmental origins of disease. Again, her full bio is available on the I3 website. Berna, we eagerly await your presentation, please.
Thanks so much, Mina. Do you see my slides now, just to make sure?
Yes.
Okay.
Yes, we do.
Okay. Hello, everyone. It's a true pleasure for me to participate in this event. I'm really thrilled, and I'm very grateful to be able to speak in front of you. As Mina kindly introduced, I recently established my lab at Yale School of Medicine. But today, with the interest of topic, I will be presenting some of my recent postdoctoral work in Magdalena Zernitschka-Goeth's lab at the University of Cambridge, UK. With this direction, I will be presenting a little bit about what we've been doing in the past few years, and discuss how stem cells can be used to model early embryogenesis within three-dimensional in vitro platforms. I don't think I need to go into much detail about the early stages of development for this audience, but I will cover this very briefly. As we all know, at the end of the first week of development, the blastocyst forms. And by this point, the blastocyst comprises three fate-restricted lineages. The embryonic epiblast inside, shown with gray, that gives rise to embryo proper, and surrounding extra embryonic lineages, trophoectoderm and primitive endoderm, that give rise to placenta and yolk sac. With blastocyst implantation into the uterus, these early lineages undergo a dynamic transformation, and embryonic morphology overall drastically changes. These changes, known as pre-to-post remodeling, lead to the formation of cylindrical post-implantation structure in mouse, whereas in human, the embryonic architecture forms into a disc shape. So the original uniform cluster of cells within the early embryo is transformed into highly organized layers of tissues by these events. Soon after this remodeling, the embryonic symmetry becomes broken to establish body axes at the onset of gastrulation, and I will be explaining these events more later in my talk. How the early embryo can undertake this complex transformation with such fidelity is still not very well understood, and this is because it is very difficult to decouple the cellular movements and more molecular events as they occur in vivo. And the possibilities to perform in vivo manipulations are quite limited. Conducting genetic experiments on mammalians is often time-consuming and also complicated by the limited accessibility of the embryos after implantation. And so these difficulties in studying early development in vivo have prompted efforts to model embryogenesis in vitro using stem cell lines, which are the building blocks of the embryo and more amenable and tractable for experimentation. There has been enormous progress in using these cell culture systems to model embryonic development, and these model systems have become very popular over the recent several years. And various independent labs produced very useful, beautiful platforms with each model system looks at different aspects of embryogenesis. This slide, I try to give an overview of some of the existing model systems, but these are mainly on mouse. Later in my talk, I will also talk about the human systems. So most of these systems use embryonic stem cells alone and try to induce developmental patterning with growth factors. Some systems, including our pioneering ones, combine embryonic stem cells with extraembryonic stem cells in an attempt to rely on the spontaneous interactions between these cell types and shape the embryo by self-organization processes. And what I also really want to highlight is that all of these existing models are unique and really useful in their own way, but they are not the perfect replicates of natural embryos with full developmental capacity. These stem cell-based embryos are relatively simple systems, lacking some features, yet effectively mimicking specific subsets of events in the real embryo. And what's really beneficial is that we can take a bottom-up approach to study poorly understood developmental events. And so one of the developmental events we were interested in when we first started this work in Cambridge several years ago now was early post-implantation morphogenesis and symmetry breaking events. Because this is the period in early mammalian embryogenesis that we understand the least due to the embryo inaccessibility once they implant into the body of the mother. And with these advances in stem cell biology, we developed the first co-culture platform for embryonic and extraembryonic stem cells, including trophoblast and endoderm stem cells to generate three-dimensional embryo-like platforms in vitro. So to mention this platform a little bit, in our design, we seeded single cell suspensions in a medium supporting the proliferation of all these three cell types. For the aggregation process, we use microvalves, as this illustration shows, which we found these microvalves are essential for proper aggregation when starting with very small number of cells. At the optimized cell density, which is about 30 cells in total, the three cell types show self-organization and form multicellular aggregates. So this is a representative stem cell structure 4 days after aggregation. And importantly, stem cells self-assemble and sort into the appropriate configuration. The cells organize such that pluripotent stem cells in red and extraembryonic trophoblast cells in blue cluster next to each other, and the positioning of these compartments defines the first axis of the structure, the proximal and distal axis. Both of these compartments become enveloped by another extraembryonic layer formed by extraembryonic endoderm, namely Zen stem cells. And this morphology mirrors that of the natural post-implantation mouse embryo. We also performed some time-lapse recordings of our 3D structures over time in culture, which is one of the benefits of this system, allowing direct observation and tracking of the spatiotemporal development. So what happens in the first few days of the assembly of these structures? We have the correct configuration of cells along the proximal distal axis, but does this configuration facilitate the correct cellular, biochemical, and molecular interactions between the stem cell compartments? The short answer is yes. Indeed, we found that these structures follow many steps of early post-implantation patterning that we normally observe in mouse embryogenesis. These snapshots show their progressive development over time in culture and the process of luminogenesis, which we found very similar of proamniotic cavity formation in natural embryos. This tells us that cells appear to be functioning as they do in vivo, and the different cell types are communicating with each other to guide the progressive change in morphology. When we tracked cell movements with time-lapse microscopy, we also observed changes in cellular polarity with distinct domains of the stem cell compartments. One cell is labeled here. In these observations, we see cell movements similar to epithelial to mesenchymal transition, and this was the first sign of embryonic symmetry breaking. In the natural embryo, we know that these events mark the onset of gastrulation. Then, to provide insight into these morphogenetic events, we also analyzed essential signaling activity, including nodal signaling in these structures. And for this, we generated them using nodal reporter embryonic stem cells combined with wild-type extraembryonic stem cells. So Nodal first showed uniform expression throughout embryonic compartment at an early time point in culture. But later on, as in vitro development progresses, this signal becomes asymmetric. It enriches on the presumptive posterior side. This shows that the essential signals become localized to pattern distinct domain within the structure. And this also indicated us that these localized signals are guided by the extraembryonic cells that are present in this platform. So as a result of these localized signals, we have observed that these 3D in vitro structures can initiate gastrulation-like events, which we found very, very exciting. This example of stem cell embryo shows the broken symmetry on the left, and asymmetric brachyury expression in green marks the mesoderm-like domain, restricted to the one side of the structure. Again, this mirrors similar events in the natural gastrula on the right. Also, this marks the second body axis formation within these structures. As mentioned before, the first axis is the proximal distal, marked by the position of embryonic stem cells and trophoblast stem cells. In the case of the embryo, these are epiblast tissues and extraembryonic tissue. And the second axis now is the anterior posterior by the formation of mesodermal domain on one side. We also used a spatial transcriptomic mapping method to compare gene expression patterns of stem cell structures against natural embryos. I'm not going to go into technical details now, but ultimately this confirmed that our stem cell-based embryos are very similar transcriptionally to the mid-gastrulate stage natural embryo. So to summarize this first part, with this stem cell-based co-culture system, several post-implantation developmental milestones can be recapitulated in vitro. This system first shows self-assembly and self-organization of the different cell types. With this, structures then undergo pregastrular morphogenesis, and later they also recapitulate aspects of actual gastrulation patterning. But I really want to mention that there are still notable limitations to overcome, and these apply to all current stem cell model systems. And first of all, it is important to improve efficiency of these in vitro systems. In our system specifically, aggregation design relies on interactions of multiple stem cells in the right number and ratio, which can cause variability. And this is a technical challenge, hopefully future bioengineering approaches will resolve. Anteriorization is also not robust in this system, which I always get questions on this. This limits the ability of, for example, neuronal tissue formation in our platform. Another point is that biomechanical features of stem cell aggregates are, of course, different than in vivo. And of course, this perhaps limits the lineage differentiation capacity and perhaps overall their developmental capacity. And which is why all existing stem cell models should be taken carefully and with realistic expectations. Again, they are not the perfect replicates of natural embryos with full developmental capacity. But despite these limitations, there are still lots of possibilities and open questions that can be addressed using these systems. So moving on to my second part of my talk, I want to emphasize that an essential feature of this system that I just talked about is that it effectively bypasses pre-implantation steps. To cut a long story short, this is because of the cell states induced by stem cell culture conditions. And so we then asked, can we also engineer an earlier developmental stage with a different 3D approach where we don't lose the stem cell potential of the early blastocyst? For this, our next approach involved using stem cells, again, mouse stem cells cultured under a specific condition called extended pluripotency. And inspired by the beautiful work from mouse blastoids by Nicola Rivron lab, we managed to form 3D structures that mimic mouse blastocysts by using our specific conditions. And here is the one example. This approach permitted efficient formation of inner cell mass, like compartment, with the capacity of forming all blastocyst lineages from stem cells, which we confirmed with single cell transcriptomics. We were intrigued to test the implantation capacity of these blastoids into the uterus, and so we transferred structures into pseudo-pregnant females. With this, we observed successful decidualization after blastoid transfers, although decidua were smaller than those induced by natural blastocysts. And through histology analysis, we found that blastoid-induced decidua was patterned to generate the primary and secondary zones along the mesometrial antomesometrial axis. And we know that this is crucial to support the implanted embryo before placental vasculature is established. To compare, naturally developing decidua is shown here in the bottom images. We can also see the tiny implanted embryo here within the decidua. However, in the blastoid implantation example, despite the progression in decidualization, blastoid development was not sustained beyond this point. We observed the breakdown in decidual luminal epithelium, which was clearly allowing implanted blastoids to make a direct contact with the underlying maternal stroma. Yet, impaired morphology of these blastoids, as you can see here, they look like crushed, even though the expression of CDX2 marks the structure in the decidua, but they look very impaired, and this suggested that resorption occurred at this point. And this indicated that abnormalities arise perhaps after direct physical contact was established within the maternal tissue. And this appears to be one of the current limits with this system. So far I talked about mouse stem cell-based models, but where is the field in terms of human-specific systems with human stem cells? While there's an ongoing effort to model post-implantation development by several labs for a few years now, very exciting new papers were published only a few months ago that shows how to make a human blastoid model. In these new model systems, the design principle differs. Two studies used human naive pluripotent stem cells, but with different culture conditions, which were from the Junwu lab and the Austin Smith lab. Another system uses human-induced pluripotent stem cells, which we will hear more about from Jose in a short while, and I cannot wait to hear. And we also had a very similar work that is now on bioRxiv, where we tested the capacity of human expanded potential stem cells to form structures and mimic some aspects of early human development, such as early polarity events to induce early cell fate, followed by cavitated cystic structure formation, and further reorganization to late stages upon extended culture. So these are very exciting for the field, and I am sure there will be lots more interesting follow-ups in the near future. So before finishing my talk, perhaps a few questions comes to mind, and some of these are, why do we all put so much effort in generating such platforms, and how all of these technologies will help us? And of course, how we can further utilize this system in human health and disease. A huge benefit is that these in vitro models can be produced in large numbers, and these systems are easily controllable at various levels, both genetically and environmentally. And thus they offer a great opportunity to study genetic and environmental factors underlying early embryogenesis at these crucial early stages of development. These benefits also make them very relevant and impactful for biomedical applications, where developmental diseases and pregnancy malformations can be modeled that cannot be investigated in the natural embryo itself. In my lab now at Yale, we are particularly interested in application of these systems to understand early embryo development in the physiological context, as well as in the pathological context of the modeling genetic disorders and origins of diseases. Our ability to accurately model these processes will be fundamental to our understanding of what makes us human and how we can treat human diseases. And with that, I will stop. I first thank Magda for her support and my previous colleagues for their excellent contributions. I also thank my amazing growing lab at Yale and my colleagues. And finally, I would like to mention that we are now hiring postdocs to join the lab. If you are interested in this research and would like to join our lab, please get in touch. Thank you all for listening, and I will be happy to answer your questions.
Thank you, Berna. Thank you very much. That was really a fantastic presentation and a great overview. So I will just ask a question, just maybe to get things going. So you mentioned that all of the models have certain limitations. So what really makes a good stem cell-based embryo model? So for instance, in terms of human blastoids, we are seeing models emerging and they all use different approaches to generate the same developmental stage. So do you think that these will differ, and what characteristics will be important in making them good models of, say, a blastocyst in this case?
Yeah, I have to say, this is an excellent question and biggest challenge right now in the field. At least I see that way. That really depends what morphogenetic step you are trying to recapitulate in vitro, and what state of cells that you are using. There are some challenges to make the very complex processes happen in vitro. I think one of the challenges, like gastrulation and post-gastrulation stages, are really nicely modeled in gastruloid systems. But if you start earlier stages, then this spatiotemporal development becomes less efficient, because you are asking more to those structures, and then efficiency can decrease even further. What will make them better? I don't have right now a direct answer for that, but it's great that field is moving forward very quickly. More approaches are appearing, and each are becoming really, really on point. And yeah, these are very exciting, and we will see what will happen in the future.
Okay, thank you very much. And now it is my absolute pleasure to introduce our next speaker, Professor Jose Polo. Professor Jose Maria Polo graduated from Buenos Aires University as a biochemist and obtained his PhD in 2008 at the Albert Einstein College of Medicine in New York. He then moved to Boston to the Harvard Stem Cell Institute to work on the reprogramming of adult cells into induced pluripotent stem cells. In June 2011, Jose established his independent research group at Monash University, where he holds appointments to the Departments of Anatomy and Developmental Biology and to the Australian Regenerative Medicine Institute. His research group focuses on the molecular mechanisms governing the reprogramming process and the epigenetic mechanisms underpinning cell fate. So it's my pleasure to introduce Professor Jose Polo with modeling human blastocysts by reprogramming fibroblasts into iBlastoids.
Thank you so much for the invitation to be here. It's a real pleasure. So I will talk to you about how we're going to try to understand the process of reprogramming to the generation of iBlastoids. So as all of you know, sperm fertilizes an oocyte, and this gives rise to the zygote that then keep developing to give rise to the blastocyst, and if continues into an early embryo, and if everything goes well, you generate a baby. And something that amazed me for all my career is that any individual is made of hundreds of different cells with different functions, different morphology, even phenotypes. However, the information to make all of these cells, all of these different cells, is without a doubt already in this zygote. And many years ago, John Gordon asked what happened with that information during development. So he took the nucleus of a somatic cell and put it in an enucleated oocyte, all this with frogs. And with that experiment, he was able to regenerate a whole organism. Yes. And with this, he demonstrated that the information is not lost during development. In other words, all the cells of your body contains the whole information to make another individual, another of yourself. Yes. And this, if we put it back now into another concept that arose at the same time in the 60s, and in this case, the landscape, the epigenetic landscape of Conrad Warrington, What we can see this is that if we get, for example, the cells of the inner cell mass, they are subject to these, what we call epigenetic forces, and they change the cells, the gene expressions, and the cells start to develop and mature and differentiate until you get the different mature cell types that will make your body. This, in the years to come, we know that the inner cell mass cells can be in vitro. We can derive what I call embryonic stem cells, and these embryonic stem cells are still having the pluripotency potential, and they are able to give rise in vitro to, in theory, any cell type of the body. And again, as I was telling you before, the whole information is contained in every of our cells. So in theory and in practice, we can take any of these cells back into this state. And this is what we call nuclear reprogramming, which is basically taking, using, in this case, transition factors to take a mature cell into another mature cell. And there is many different reports of how to do this. For example, in this case, B cells into macrophages, or the most famous one probably taking a fibroblast into an induced pluripotent stem cell. This is work by Jinya Yamanaka and Takahashi, where they demonstrated that you can take a human fibroblast and reprogram back into this pluripotent state, which basically in many ways behave like a embryonic stem cell. And this has revolutionized regenerative medicine, because the promise of this reprogramming research is that we can take a cell of a patient and reprogram into a pluripotent state, and from that pluripotent state, we can now differentiate it in vitro into any cell type, for example, a disease cell type if the patient has a disease. And with that disease cell type, generate disease models, which allow us to do drug screenings in order to solve, to find new therapies, or even to generate cell therapies directly to be able to cure that patient. And this is a reality, actually, and is many new drugs are coming out of this type of research, and also there is several cell therapies derived from iPS cells now entering into clinical trials. So my lab was, or is, interested in understand how this process happens. How, for example, a human fibroblast become a iPS cell, a pluripotent cell. And we decided to study this in two stages of pluripotency: the naive pluripotency state, which is reminiscent of the pre-implantation state of pluripotency, and the prime pluripotency state, which is reminiscent of the post-implantation state of pluripotency. So all these studying in vitro. So basically what we did is we took fibroblasts, we started reprogramming with the 4 transition factors that Yamanaka discovered that are capable of reprogramming a cell into pluripotency, called OCT4, KLF4, SOX2, and MYC, or KSM in short. And we reprogram fibroblasts all the way into prime iPS cells or naive iPS cells. And when we were designing this experiment, we designed an experiment where we did single cell RNA sequencing or single nucleus RNA sequencing. Basically, we take the cells at different intervals, and we look at how every individual cell is changing, how the RNA is changing during this process. And by doing this, we were able to unveil the different pathway, all the trajectories that the different cells take to go from a fibroblast, in this case in black, into this prime state down here. So all these dots are different cells, and this you see the cells coming at different time points coming here, or cells going all the way into the naive state of pluripotency. And in this paper, we discover all these trajectories and which regulatory elements have to be activated in order to do this. But we also noticed that when you took the trophectoderm signature and you ask the system, is the trophectoderm signature expressed in any of these cells? We found there was a group of cells that they were expressing these trophectoderm associated genes at a high level. And it was very surprising because reprogramming is trying to create cells that emulate the epiblast of the cells, no? The pluripotent cells. So we decided to check these cells, and we culture the whole cells at this time point in a media that it was derived to generate trophoblast stem cells from blastocysts or from first trimester placenta. And when we did this experiment, we were able to derive what we call induced trophoblast stem cells. And these induced trophoblast stem cells had all the characteristics of trophoblast stem cells, the one derived from blastocysts. They were positive for the classic markers of these cells. And more importantly, they were able to differentiate into the main cells of the placenta, that are the syncytiotrophoblasts and the EBTs here. Furthermore, if we put the induced trophoblast stem cells in a mouse and we collect the urine is able to give an HCG pregnancy test positive and out of the counter test, because they are secreting, and when they differentiate in vivo, they are secreting HCG, and this is detected by the pregnancy test. So this, we published this last year, and meanwhile we were finishing this paper, we looked better to the cells that we were generating during this reprogramming process, and we realized that by day 21, actually, beside the epiblast-like cells that they were generated, beside these cells that upregulated the trophectoderm signature in some way, we found cells that were also upregulating the primitive endoderm, yes, or signature in during reprogramming. So we wonder what happened if these cells were putting together and interacting, what happened with these genes, and so we took the cells during reprogramming again, and after 21 days of reprogramming, we aggregated so they could contact and signal to each other. And to our surprise, they self-assembly in 6 days into these three-dimensional structures. And when we started to analyze these three-dimensional structures, and we realized that they were they have a cavity, they express nano, for example, and they actually, when staying with other markers, we could see, in this case, actin, allow us to see very well the morphology of these cells, being the cells in the periphery, and this more epithelial type of morphology. Meanwhile, the ones in what is this inner cell mass-like structure were more globular. And in retrospectively, we are calling this now iBlastoids. There was many experiments that allow us to come to the conclusion that these were blastocyst-like structures, so we call it iBlastoids. So when they are staying with classic markers of trophectoderm and epiblast, as we can see here, the cells staying with CDX2 and in the periphery layer, and they staying with nano in this inner cell mass-like structure. It's not just CDF2. We can see, for example, that GATA2, another classic trophectoderm marker, also marks all the cells in this outer layer. And we can see, for example, cells expressing SOC17, which is a classic marker of the primitive endoderm. We also have stains for GATA3, for example, and SOX2 and OC4 in this. So we continue doing the experiment by taking the cells and doing a single cell RNA sequencing. And this, when we look at the cells on these blastoids, we could see that they were expressing OC4, for example. Many cells express CD2 in different regions, and SOC17, as I showed before. And if you look now at a signature that is a more stronger way to see all the cells, because single cell transcriptomics has this problem that it has a lot of dropouts, which means that some cells you cannot detect a gene, and this is common. But when you look at a signature that is composed of many genes, you can see better where, for example, the epiblast, the cells that have a regulated an epiblast signature are, or the trophectoderm, or the primitive endoderm. Furthermore, when we integrated this with single cell dataset from blastocysts, from blastoids generated by IVF, from 2 different groups, and we could see that in what is called a UMAP analysis, when we integrate it, we can see that our, for example, here, our primitive endoderm-like cells in the light green, they occupy a similar position, similar area that the primitive endoderm cells from blastocysts. The same happened with epiblast-like cells or with trophectoderm-like cells. Furthermore, if you do a classic correlation analysis between the different lines, you can see that the correlation is very high between our primitive endoderm, epiblast-like cells, and trophectoderm-like cells. We also found that these structures have other types of cells that we don't know very clear what they are. Yes, they have some what we call intermediate signatures, so they have a little bit of this and that. It's yet to be derived. So as we know, embryonic stem cells can be derived from blastocysts and trophoblast stem cells from trophectoderm. Of course, Yamanaka demonstrated that iPS cells can be derived from fibroblasts that are analog to embryonic stem cells. And I show you just now that induced trophoblast stem cells can be derived also from fibroblasts by reprogramming. So we wonder if we could actually derive these lines, these naive pluripotent stem cells or prime pluripotent stem cells of trophoblast stem cells from blastoids. And we call it BPSCs or PTSCs. And yes, indeed, you can take these blastoids and generate naive pluripotent stem cells or prime pluripotent stem cells or trophoblast stem cells. So from that perspective, these iBlastoids have the capacity to generate all these stem cell lines. And furthermore, we subjected the iBlastoids to an attachment assay that is used to study with blastocysts, is used to study the initial, to model the initial state of implantation in vitro. So this is a very kind of simple assay where the blastoids or the blastocysts, or in this case the blastoids, are attached into the surface, and we let them grow for up to 4 days. And what we found is that very rapidly, this is in less than 4 days, in 4 days, you have an expansion of the trophectoderm into the trophectodermal cells expand and grow. You can see in CDX2, but we have with other markers that we show now. And the same, there is a small expansion of the epiblast. And you can see, for example, how the primitive endoderm also advanced, separated from the epiblast, what is has been reported in blastocysts. If we look at other training, you can see how clear the trophectoderm-like cells progress into a differentiated trophoblast. For example, we can see how the cells become keratin 7 positive and pretty strong around, and they express, for example, HCG again. And if you look at, if we use a facting to see better the morphology, we can see this globular, again, the globular morphology of the epiblast. And we can see, for example, already we can detect cells that have the morphology and the binuclear of the syncytial trophoblast and extravilus trophoblast. And we also detected the generation of what could be called a proamniotic-like cavity. Yes, in this attachment assay. This again has been described with blastocysts. Here you have, and actually occupies a three-dimensional aspect here. So you can imagine we are very excited about this iBlastoids and all the possible application and the capacity to model this initial 6 days of between day 6 and day 10 of human development. Yes. And for example, we think that they could be used for, of course, understanding early human developing to improve IVF techniques and even for IVF training, which I hope that we can discuss today about it. Developing new genetic screens, since they can be generated in big quantities. In just in one experiment, we can easily generate hundreds. So we think that this will allow us to use it for toxicity assays, for example, or toxicity assays in, for example, in placentations, if we can model those initial 5 days, 4 days of placentation. So we hope that that can lead to improved techniques or methods or therapies for improving placentation. So I would say thank you to all of you, and remember, basic research matters. Yes, when we started this journey, we wanted to understand how this important process now in biology that is called reprogramming happens, how it works, and we end up finding a way to generate first induced trophoblast stem cells, which allow us to model the first placentation in vitro, and then we even found a way to we came up with this way where we can generate this iBlastoid that is a great model, although with some deficiencies, but a good model of the first stages of human development. So thank you, and this work has done by the people of my lab with collaboration from many people, especially collaborations with the laboratory of Ryan Lister and Owen Rackham and Amanda Clark. This work was, the first part of the work was done by Ethan and Fernando and Christian, and the second part by Ethan and Gia with, of course, the help of the entire lab. So thank you so much, and I will now answer any question that you have.
Thank you so much, Jose, for this fascinating talk. I have to make an admission before I ask you a series of questions that came to my mind. I'm a clinical embryologist, so as a clinical embryologist, it's sometimes difficult to understand the details of talks like yours and Berna's when it gets into some of the molecular aspects, for example, of your work. But to me, I mean, it's just fascinating to see how you have gotten to this point, and it's kind of difficult to imagine the steps. So, for example, I wonder, when I think about stem cells, I'm thinking about these monolayers of cells growing on a dish or a flask of some sort. How do you go from that to this structure, this 3D structure? And what's the efficiency of the formation of these structures? How many of them do you get in a culture? I noticed that you had a drawing of a container or a dish that is sort of I don't know what it's called. It's not, conical. And I wondered, is that how you grow them?
Hi, thank you. And thanks again for the invitation. So we grow the cells monolayer, yes. And actually, in this particular case, we grow the cells, we are growing fibroblasts. And then when we are reprogramming, they still monolayer. And the reason that we did the experiment was because when we noticed that these signatures were getting upregulated, we decided to, as you well said, in a monolayer, they don't interact well because they are sparse. So what we did is using these plates that are called agriguel that they have been used in the past or in mouse to do blastoids, or also in other methods to make cells interact, because they are like, we call it inverted pyramids, yes, and they allow the cells to come together. So we just did it so they could interact. So what we do is, yes, we have the monolayer, and when these signatures appears during reprogramming, they don't just, to be clear, they never make it into what you will call a stem cell by that point, yes. Of course, they have regulated a lot or a big part of the transcriptional signature, but we will not say that they are yet in stem cells. Actually, we don't do the experiment in pluripotency media, so the cells are not stabilized by that media yet. But anyway, if you trypsinize or just to make it simple, the layer, and then you put it in those cones, in those agriguels plate, with a well of that agriguels contain 1,000 of these micro agriguels, yes. So, and when we put the cells there, we put in around 100 cells per agriguel, that's the count. And out of those 1,000 agriguels, depends experiment, you will get around 100 to 150 blastoids. And we haven't played yet with trying to make it more efficient at all. Like, so this is without trying to make it more efficient, that we know that there is probably, we can increase that efficiency, but we haven't played with that yet.
Very interesting. I have, if I may, Mina, I'll just ask one more. So you mentioned that we could potentially use these for IVF training. That's very intriguing, because we're always in need of material to use in training, and of course research, but especially training. So are you able to vitrify and warm these blastoids successfully? Because that would be a key.
Yes, I completely agree. We haven't done that experiment. The reason that we haven't done, but I will explain why. The reason that we haven't done those experiments yet is because in Australia, when we make this discovery, after basically all the information, all the experiments start to come up back to us, yes, and analyzing, and we realize that they were blastocyst structures. And by that time, we contact what is in Australia the NHMRC embryo license, and we told them what we have made, and for that we need a license, and we are in the process of generating the license. So, in order to keep doing those experiments, so yes, I think that the first experiment that we will do as soon as we can and start doing this, we will try that. Exactly. Yes.
Fantastic. Mina, would you like to? I have more, but why don't you go ahead?
Thank you. Yeah, there are definitely many questions, but a question, I guess, to both the speakers is you were touching a bit upon the regulation. So I was just wondering, do you think that there are ethical issues related to this work? And especially what comes to mind for me is that these are in effect clones of the cells that they are derived from, these blastoids or the structures that you are forming. So how will this be regulated in terms of reprogramming? So the donors that are giving their fibroblasts, or the cells that you are using, or in terms of the embryo from which these cells are being derived and then being formed into an embryo again. So yeah, I'm just curious as to what your thoughts are on this.
Berna, do you want to answer or you want me to go first?
I was waiting for you to take the lead, but I can go as well.
I can give it a try. The first, I will do a couple of clarifications that Berna may have thought as well. First of all, they are not embryos.
Yeah.
Yes, they are models. Yes. So from that perspective, at least, we don't see it as we are creating an embryo. We are creating a model. Yes. In the same way that we were creating before more simplified models. Yes. And yes, you can consider that you are cloning those cells in the same way that you are defining cloning there as a cloning of the cell, of the nuclear information. But when you create iPS cells, you are doing the same thing. You are doing copy. So that's not. So of course, now the ISSCR, that is the International Society of Stem Cell Research, has released the new guidelines to try to regulate all this work. And they, in the new guidelines, they propose a guide, I guess is the word, that when doing this type of research, you need to obtain what is called an embryo ethics approval. So basically, you need to get your ethics approval according to the national laws, of course. But this ethics committee that will regulate this type of work with these embryo-like structures, what the ISSCR call it integrated models. They require kind of more sophisticated ethics approval, where the ethics panel has embryology knowledge and knows how to handle the type of research. So basically, you go through the same process that if you were to work with blastocysts, yes. But it makes also a very clear difference in the sense that some rules that apply from blastocyst don't need to apply necessarily to work with these models. Berna, do you want to add something else?
Sure. Yeah, I was gonna start kind of with the same things. Yeah, these are what we need to understand. These are just model systems. These are like they don't have full developmental capacity, or they don't have organismal potential. We are creating these to understand specific subset of events that occurs in vivo. But I mean, again, like they are not going to, they don't have organismal potential. I think we as scientists, we have a great task here to put realistic expectations to other scientists or to public, and this is like how we present them, how we name them, how we introduce them to many societies. I think that's very important. Ethically, that's very deep issue. I don't want to really get into that part, but what I can say, ethically, if we don't investigate developmental events, that's equally unethical, because we need to understand these processes. And these models, again, they are just model systems to investigate some parts of the development.
Berna, can I ask you, you repeated a couple of times that they don't have organismal potential. Can you expand on that?
Yeah, so what I mean by that, so if we look at the blastoid systems, for example, they initiate implantation dynamics, but they don't implant. They don't further develop in utero. Or if you look at the other model systems that recapitulate gastrulation, those are mainly just embryonic stem cells, only embryonic stem cells, effectively forming germ layers. But those structures don't have placental support or anything that would allow them to go further and go into fetal stages, basically. So this is why I'm trying to emphasize that these systems are, it depends what window you are interested in. You can use them to understand how germ layers are being specified, what signals are important to specify germ layers. But if the question is, how am I going to make a fetus? That's not what this research is, and that's not what we as scientists trying to make.
So when you looked at implantation, this was in vitro?
Yeah. No. So in our mouse blastoid system, and also the other lab, Nicola Rivron's blastoid system, we tested their implantation capacity by transferring them to the pregnant mice. They do initiate some initial steps. They do interact with endometrial tissue, but shortly after, we don't see development. Yeah.
I mean, this leads to.
Can I clarify something? Sorry, I don't want to interrupt. But no. I think that it's super important in this moment to clarify this. So the work that Berna is talking about, which by the way, I'm a super fan of your work, Berna. Like lovely work, yes. The work that he's talking about is with mouse blastoids.
Right.
Yes, human blastoids, neither the iBlastoids or neither the blastoids generated by Jung-Woo have been put into a uterus. That's actually prohibited. Yeah. Yes, to do that experiment. Yes. So and when we talk about, that's why we talk about attachment, because it's an in vitro attachment, and it models, as a model can be good, the peri-implantation phase. Yes, but it's all in vitro. But what Berna is saying is that even when you do in mouse and you put in a uterus, initiate the movements of the first stages, but doesn't continue. Yes.
Right. Thank you for that clarification. Understood. If when we look at these blastoids, they look similar to day 6, 7 sort of hatched blastocysts. Can you tell us in both systems how long does it take to generate these, and when you do the transfers, I mean, is this equivalent? And I mean in the mouse, and then of course the attachment in the case of the human blastoids. Are these equivalent to a day 6 or 7 human blastocyst or mouse blastocyst?
Berna, you go first.
So for mouse, what I can say about our system, we did do some transcriptomic analysis with single cell sequencing, and we do see their transcriptional signature is most similar to late stage mouse blastocyst, around 4.5 after fertilization, which is around day 6 human blastocyst. With the human blastoid, with the model we were trying to we were working on, what I can say, these expanded potential cells comes with many issues, and what they can do is, what we found is actually very limited. They do differentiate to some extent some trophoectoderm cells, but they also have some transcriptional signature which is not existing in the real human embryo. So it's like a bit of, how can I put that? It's a bit of, to some extent, yes, we do have a transcriptomic signature, but it's not exactly mirroring what we see in the natural blastocyst.
So with the iBlastoids, we did that experiment as much as you can do. So we compare the specific, and we use our single cell transcriptomics and to compare with different transcriptomics of blastocysts E5, E6, E7. And what we found is, yes, is more similar to E6 and E7. However, you find and also cells that are more similar to E5. But all these, you need to check it over with a pinch of salt, because you are comparing basically one measurement, of course, of multiple genes at the same time, and you are trying to do just a correlation analysis with a similar measurement in the case blastocyst, yes. And all depends how the original blastocysts were staged. Yes, that we didn't stage it because we don't work with human blastocysts. So, and the techniques were different also. So you need to check this comparison with a pinch of salt. But yes, we could say that they match a 6-7 blastocyst in many ways. And as well as I showed before, we also found cells that that have a mixed signature that we need to still trying to find out what are they doing there, basically.
Mina, would you like to pose some questions from the audience?
Sure, yeah, there is a question from the audience from Richard Rollins here from Illinois in the United States. He asks a question for Berna, and he asks, what media are you using to culture the blastoids? Could the blastoids maybe serve as a cell source for proofing future culture media for IVF or ART?
Yeah, that's a great question. So the protocol we use currently to grow them, it's based on more like how we grow stem cells, rather than using like IVF based or embryo culture based media. We use like standard DMEM, and we supplement it with like many nutritions. We did try in some preliminary experiments, we did try using for mouse blastoid, I'm talking now. For mouse blastoid, we tried KSOM, which is normally how we in vitro culture mouse embryos. And surprisingly, trophoblast stem cells did not form trophoectoderm. They didn't cavitate. They were just making bolts of clusters, basically. I don't know why that was different, but I'm guessing because in the stem cell culture media, we are supporting what trophoblast stem cells need, like FGF4 or heparin. We are giving these factors. So these are helping, of course, to trophoblast stem cells to behave. In the human blastoid, we did use IVF media to a portion, not full portion. It was like half stem cell media, half IVF media. We did a mixture, which we found, interestingly, it's promoting cavitation. It's helping for the cavitation. Without using IVF media, we found it doesn't help with the cell identity, but we found the stem cell structures were just cluster of cells, not cavitated structures.
Okay, and another question from the audience for Dr. Polo from Juanjo. Friday, he says, I was wondering what is the proportion of iBlastoids that can be successfully generated? And from these, what is the proportion of properly transcriptionally differentiated cells they have? So in other words, how many cells have to be differentiated for an iBlastoid to differentiate properly?
Okay, so if I understand so well the question, you generate, when we do it in these agri wells that they have in a, let's say, you have in a well, you have like these 1,000 agri wells inside. All the agri wells will get a structure, yes, and you get different type of structures. And again, and we based on our quantifications, around, depends the experiment, around 5% to 15% will have a structure that you will deem as a blastoid. So I would say around 10%. Mm-hmm. This is without working in trying to make it more efficient. Of course, we haven't grabbed the, let's say, 1,000 of them and do single cell transcriptomics in the specific 1,000. But when our transcriptional data comes from grabbing 200 of them, actually this was done in replicate, and there where we see. So there is definitely a lot of space for improvement here, yes. But yes, the short answer is around 10%, I would say, for the moment.
Do you have any idea, either with the mouse or the human blastoids, what's the degree of homogeneity or heterogeneity among the different blastoids? So, you know, in the mouse, they are pretty homogeneous. You culture, you know, they all look the same, they all do the same. Whereas human embryos are very variable, even within the same cohort of eggs. So do you have any idea about that?
So, please, no. I thought that because yours are super homogeneous, the answer was like, thank you. Go ahead.
Yeah, well, I'd say these stem cell structures are as heterogeneous as they could go. Each structure is, yeah, I mean, to be honest, we've never really looked into this deeply, which we should, but I have a feeling based on our experimental results, the initial population of cells, so we do the seeding on these agar plates, which has 1,200 microvalves inside a well. So then cells basically seed into each microvalve. We don't know who is coming where, what is the potential of that cell in each tiny microvalve. And I think this is really affecting the ultimate structural formation. When we do IF stainings or transcriptomics, yes, we do see variances from one structure to another. Specifically for blastoid, I personally realized primitive endoderm formation is very heterogeneous from one structure to another. Mm-hmm. So they show differences.
And in our case, so one of the reviewers asked us to do a clinical grade of the structures. So we give them to somebody that had that experience, yes. And basically, what we found is that using the good, fair, and poor grade, yes, we found that in average, they were what you will call fair, yes. But we have, and I'm looking at the thing now, we have very few that were deemed poor, and then I will say like, I don't know, 25% that were deemed as good. So we have good, fair, and poor. You have a distribution with the fair in the middle. All depends which, because we or she did score the ICM and the T separately, yes. So the ICM, we have like a distribution where the center, let's say, is between fair and good, and then, or a score of 1.75, yes, and the T has a similar score, 167 in average, yes. And so I know that answers your question. But then, yes, we have, if you use immunostaining, you see different things. We always see to do all this depending, of course, we picked the ones that were cavitated, yes. And when we do that, yes, we see the trophectoderm and the inner cell mass and the stain for that. But yes, there is, for the moment, we have, it's heterogeneous, yes.
Would be interesting. Yeah.
Yes. Basically, the people in the lab was able to separate them pretty fast, yes, and you will get like hundreds of structures that are more or less homogeneous after that.
Yeah, I'd like to add here, there is a good interest to use bioengineering platforms, bioprinting platforms, where you can control how many cells are coming together, or you can sort the cells with the high stem cellness, which could help increasing efficiency. But there is a caveat on that. Yes, you can increase the efficiency, you can fabricate so many of them with very similar identity, but then the beauty of these synthetic embryology platforms are, embryos are teachers, but these systems are doing various things. Then you can understand how it goes awry. Then you have a chance to address, when you take a impaired morphology, then you can understand what happened here. So I actually think really depends what you want to use them. If you need a really high efficient system for drug screening, for example, then I think bioengineering platforms will be very, very useful platforms. But if your interest is more like, I want to understand what is fundamental dynamics, I like this heterogeneity, then we have chance to address where is this heterogeneity coming from.
It would be interesting to see if some of the different types of abnormalities that we see during blastocyst formation, especially in the human, you can mimic or you see during these blastoid formation. For example, multiple cavitation, or do they actually contract and re-expand, or no?
So actually, we couldn't do that experiment. We were asked that. We don't have a zona pellucida, and
right.
So at least as far as I understand, there is some, or a big part of the field think that that's one of the reasons that you can have it. So, but we haven't done, yes, and that is in the plans, definitely.
You need to put them in a zona and see what they do. They're able to.
And along these lines, how much pressure is there from the culture itself? I mean, if we're saying we're going to model certain abnormalities that arise in vivo and so on, I mean, there is a lot of culture pressure, we know, in terms of stem cell culture in itself. So how would you control this in the future, and how relevant do you think this even is in this context? Or I guess depends on the context, but.
Yeah, it really depends on the context. Like for modeling genetic disorders, which I really have great interest on that, but I also believe like when you have so much heterogeneity in your in vitro system, then it becomes difficult. Is this coming from just the culture-induced problems, or is this really relevant to the phenotype you are trying to address? Again, I guess in that terms, having a bioengineering platform where you know your efficiency is really high, you have a really effective control, then it becomes much easier to target a gene and address what is the outcome, or like any other question comes to mind. Yeah.
Yeah, I agree. No, it's all about the interplay between what's going on with the epigenome of the system and, of course, the media that is around.
Yeah.
And I think that in all these models, that becomes super clear in the sense that if you start with a stem cell, in all the methods that are in stem cells, they are just changing the media and patterning them and changing even the 3D aggregation capacity, but you change just the media. In our case, we also go directly to the epigenome and trying to change it, but also we change the media at the same time. And so I think that it's never more clear that what a structure is, or it completely depends on in which media they are. And probably if we were to find that, at least this is how I see it. So this, as Berna said, this model, these few days of development, yes. And if we were to find other medias or other conditions that model other stages, well, we will not call it them blastoids anymore, because now they are modeling another part of development, yes, which is completely dependent on all those views that receive. So, yes, super important.
So, we're going to have to wrap up very soon, but I do want to get to this one question from the audience, which is a tough one, and sort of touches again back on the ethics of the whole work. This is from Zuzana Holukova, who says, could you exclude the organismal potential of human blastoids without actually trying it, which would be ethically unacceptable? It sounds like it cannot be disproved, and it's an ethical trap. What do you think?
So
can you disprove it without transferring,
yes,
or without?
Yes. So under the same type of question, yes, and we cannot disprove that, because what she's saying is that the experiment cannot be done. So definitely, yeah. So we cannot disprove it, yes. And so I would say based on all the evidence that we have, yes, and that was the only thing that we can do, working evidence-based, yes. There is no reason to suspect or think that this can be done, yes. And so
since you're trying to improve the models, and that's what you were talking about, the limitations and addressing these limitations, do you think at some point there will be a risk that these may have developmental potential, even though, of course, that's not what we're aiming for? But if you're improving them to the point that they represent the in vivo embryo as best as they can, where do we draw the line?
So, but basically that will be to make another model. So what you are saying is, should we make a model that can model the entire embryogenesis, which will become a completely different model of this? You can see this even better, I guess, with both systems, with like the blastoid derived from stem cells or the blastoid derived from fibroblasts like us. And in our case, 21 days later, they were fibroblasts. The only thing that we did was change the media. So, and you reach until one point, which is this blastoid that has this capacity to model X and Y. If you want to do something else, it is to model the entire embryogenesis process, you will need to generate other model. So the discussion, I guess, that will be, should we be trying to do those models? Yes, which is different from these models. In the same way that our models are different from a trophoblast stem cell that models only the trophoblast side, or induced pluripotent stem cell that only model the epiblast, some of the epiblast aspects. And the same when you come from an embryonic stem cell or a naive stem cell or an extended potential stem cell, and you start patterning like Berna probably talk, you you are just changing the media. So again, it's a matter of what model you want to do. For the moment, I think that, Berna, I don't want to talk for you, but I think that what you said, we are doing models that can give us information on these few weeks. If we were to do other models, the discussion will need to be in those models, not in these models.
Yeah, but for the later stages, it's such a complex process in embryology. It's not only embryonic tissue, extra embryonic tissues and everything. It's very, very, very complex process to even think about attempting to model any of those processes, in my opinion. And I mean, it's difficult to say what's going to happen in future, of course, but at the moment, if science comes to that point at some day, I don't know, it's a very, very deep conversation that scientists should have and draw the line and make sure this is hand in hand between scientists and also public, and in general, there will be a line for everyone, I'm sure, if it comes to that point.
Well, on that note, looking to the future, I'm going to start wrapping up the session. I want to thank you so much, Berna and Jose, for sharing your beautiful, outstanding work with us today at this I3 session. Thank you, the audience. Thank you all for taking the time to participate in this session. Thank you for your questions, and sorry if we didn't get to all of them. We got very excited with our own questions, maybe. And many thanks also to the organizing committee of this initiative, Jacques Cohen, Peter Nash, Giles Palmer, Thomas Elliott, Shaista Sadruddin, and Marianne Cervantes. They also managed the Q&A session today. We want to thank them. There is so much work that goes on to organize these sessions. So we want to thank them sincerely. I want to now hand it over to Mina, who will close the session.
Thank you very much, Mina, and thank you very much to the speakers. It was really an exceptional session. Definitely enjoyed it very much. So thank you. And of course, thank you to all the organizers. This session and the presentations will be available on the website very soon. Don't forget to rate our lectures. It is very easy. And your certificate of attendance will be sent before the commencement of our next session. Our next session is on Tuesday, as always, at 3 p.m. Eastern Time, 8 p.m. UK time, and 9 p.m. European time. And the next session is sponsored by Cook, and it's all about the embryo in culture. And we really have a great lineup. Kay Elder, Yves Menenzo, Jason Swain, and Roger Sturmley, just to name a few. So be sure to join us then. And thank you very much again for joining us today. And we hope to see you all very soon.
Thank you. Bye-bye, everyone.
Thank you.
Bye.
Bye.